Hello everyone. Welcome to the session of Biotechnology Principle and Processes NCERT exercise. Disclaimer. And today we are going to study about the Biotechnology Principle and Process chapter NCERT exercise. So learning objective is we will explore, understand the fundamental concept of the chapter 9 Biotechnology Principle and Processes by working through the NCERT exercise. So by solving the NCERT exercise, we will able to understand the fundamental concept of the chapter Biotechnology Principle and Processes. And index part, that is, we are going to solve question 1 to question 12 from the chapter Biotechnology Principle and Processes. And the first question is, can you list 10 recombinant proteins which are used in the medical practice? Find out where they are used as therapeutics. This is the question coming from the NCERT section 9.1, right? Let us see the answer of this question. So this was our question. Can you list the 10 recombinant protein that we are using in the medical practice? Let us see the answer. So before going to the examples of the 10 recombinant protein, let us first understand what is recombinant DNA technology. Recombinant proteins are obtained from the recombinant DNA technology. This is the technique that alters or manipulate the DNA, right? And this manipulation is done to get a desired type of protein, a desired type of result from the altering the gene, right? So here we are getting this recombinant type of protein that we are going to use for our medical practices, right? Through medical recombinant DNA technology. So RDT involves the transfer of a specific gene from an organism to another organism, right? An altered gene is transferred to from one organism to the another organism. This we do in the case of recombinant DNA technology, that is RDT. So transfer of one gene carried out by the vectors and the restriction enzyme as a molecular tool. So these are the two very important things. Very important things in the recombinant DNA technology. That is the vector. The first thing, that is the vector. You required vector to incorporate a desired gene in the vector, right? And when vector multiply, that desired gene also get multiplied, right? And for this, the incorporation, taking out the desired gene from an organism and incorporating it into a vector, you require molecular tools as an enzyme. Here, the restriction enzyme is working as a molecular tool. They cut the DNA at a very specific point, and every this restriction enzyme is having such a restriction site at which it cuts. So you cut the desired gene from an organism, you cut the vectors, right? And the desired gene is incorporated into the vector get multiply, and that desired gene which is incorporated into the vector also get multiplied. Now what happened after that? So here are some of the recombinant proteins that are used in the medical practices. So here you can see recombinant protein. In the column it is given recombinant protein, the name of the protein that is modified with the recombinant DNA technology and uses in the medical practice. What is the use of that, right? So the first one is human insulin. Human insulin is nowadays it is produced as a recombinant protein. Now you see that every on and off person is taking that insulin medicine, because of the diabetes, because they are the diabetes patient, the disease, right? So they have to take insulin. So this human insulin is now produced on the basis of the recombinant DNA technology. Now, what is the use of this? Used to treat diabetes mellitus, particularly type 1, right? Diabetes mellitus, in which the glucose is there, but the glucose do not get enter inside the body cells. They do not get enter inside the body cells unless and until there is a presence of insulin, right? So this glucose get encapsulated in the insulin, and then only it get enter inside the cell, and then through the respiration process it produces energy. Otherwise it will be present in the sugar only, blood only, right? Next is the recombinant protein is the example of coagulation factor 8. Factor 8. This is used to treat hemophilia A. Now, hemophilia person, in that there is a problem of getting the blood clotting when there is a cut or any damage or any wound on the skin, right? The blood is continuous flowing out because of the absence of this coagulation factor 8. Now, with the help of recombinant DNA technology, this protein can be synthesized, right? Coagulation factor 8. And this is used to treat hemophilia A. Now, similarly, the coagulation factor 9 is used to treat the hemophilia B. Next example is the erythropoietin. Erythropoietin. This is also is made nowadays with the help of the recombinant DNA technology, and it is used to treat the anemia. Anemia, the people who are suffering with the anemia, especially in the patient with the kidney failure and undergoing chemotherapy. So these are some of the very, very important recombinant proteins that are used in the medical practice. Now, the fifth one is interferon alpha, used to treat hepatitis C and certain cancers, right? It is used to treat the hepatitis C and also in the some of the cancer disease. In the question, it is asked about the 10 recombinant protein. Here we have seen the 5. Next 5 is, let us see that. Okay. Next 5 is interferon B. Interferon B. It is used in the treating multiple sclerosis. Multiple sclerosis. 7th is the human growth hormone, HGH, used to treat growth disorder, including dwarfism. You must have seen that there are some people who are not having this human growth hormone and they are having the stunted growth. They are not full grown or the normal height, right? So they are showing that dwarfism phenomena. So by taking this human growth hormone externally, right, which has been developed from the recombinant protein, they can get rid of that disorder, or they can treat that growth disorder like dwarfism. Next is the tissue plasminogen activator, TPA. This is used to dissolve the blood clot. Sometimes there is a blood clot in the body, and in cases of the heart attack and stroke, these blood clots are there. So to dissolve that blood clot, some protein is required. That is the tissue plasminogen activator. So this is also developed by the recombinant DNA technology. 9th is streptokinase, very, very important recombinant protein, used to dissolve the blood clot but less frequently than TPA. Okay, these two are the used for the dissolving the blood clots in our body. 10th one is the hepatitis B vaccine, very important, used for the prevention of hepatitis B infection. So these are the some of the recombinant protein that are developed with the help of the recombinant DNA technology and the medical practices you have seen. Next question, Make a chart with the diagrammatic representation. Show the restriction enzyme, the substrate DNA on which it acts, the site at which it cuts the DNA, and the product it produces. So you have to show the diagrammatic representation for all these activity. Let us see the answer of this question. Now, we are taking an example of the enzyme that is called as EcoR1. EcoR1. Escherichia coli. Eco means E is for Escherichia and CO for coli, right? So this was the first restriction enzyme that was discovered in the E. coli bacteria. And that is why it is called R1. Now, they specifically recognizes the sequence. Now, you have to memorize this sequence. 5' GAA TTC 3'. This is the site at which this EcoR1 goes and give a cut. It always recognizes this sequence. And if there are 100 sequences like this in any DNA genome, then it can produce 100 segments. Because it goes and cut at these segments only. It do not cut at any other segment. It only recognizes this segment. So cleaves the DNA backbone between the G and the first A. G and first A. So G at the 5' it is present, and the next nitrogenous base is the A. So it cut in this position. Produces a sticky end and 5' overhang at AATTC. So here you can see in the diagram, this is the restriction enzyme, and here you can see it make a cut at this position with the first fragment, and the second fragment segment of the DNA, right? It will cut here. So these 2 points, it will create a cut, and here you can see this is the sticky ends, AATTC in this position and CTTAA in this position. These are the point at which it gives the cut. You can see the arrow 5' to 3' DNA strand and 3' to 5' the DNA strand. So it always cut between the A and G, right? Next question. From what you have learned, can you tell whether the enzyme are bigger or DNA is bigger in a molecular size? How did you know? A very, very important question. So whatever you have learned so far in the biotechnology, biomolecules chapter, so can you tell whether the enzymes are bigger or DNA is bigger in the molecular size? So let us know the answer of this question. Enzymes are smaller than DNA. Enzyme is a protein. The nature of the enzyme is protein, right? And they are very, very smaller than DNA. So this is because the DNA contains the genetic information for the development and the functioning of all living organisms. So DNA is the biomolecule that contain all the information that is going on in your body. So there may be hundreds of protein that get synthesized in our body, and that the code, or you can say the gene for those particular protein are present on where? They are present on DNA. They are present on the DNA. So it contains all the genetic information, right? That is going to be functioning in the living organism. So it contains the instruction for the synthesis of proteins and DNA molecules. Yes, the DNA which is existing in the cell, it contains all the instruction, all the information that is required for the synthesis of protein, that is required for the synthesis of a new DNA molecule. Because you know, from DNA, the new DNA molecule gets synthesized by the process of replication, right? So For this, where all these instructions coming from? All these instructions are coming from itself, the DNA molecule. On the other hand, enzymes are protein. What is the chemical nature of enzyme? The enzymes are made up of protein, right? And proteins are synthesized from the small stretch of DNA known as genes. So there are so many proteins that are present in our body. I can take an example, that is suppose alpha amylase. Alpha amylase is an enzyme that gets synthesized in our body, and that is an enzyme, right? And for that, the gene is present on our DNA. So from DNA, first the RNA gets synthesized, and from RNA, the enzyme alpha amylase gets synthesized, right? First, there is a process of transcription, then there is a process of translation. In the process of translation, the enzyme get synthesized, right? So protein are synthesized from the small stretch of DNA. So which is bigger? Yes, there are so many genes that are present on DNA. There is not only one enzyme that is present on the DNA. There are numerous numbers of enzymes that get synthesized in our body. There are so many structural proteins that are present in your cell. That gene also present on that DNA. So obviously the DNA molecule is larger than if you compare it with any one enzyme. So these genes are involved in the production of polypeptide chain. Yes, the enzymes are made up of polypeptide chain, and from these polypeptide chain, there can be a number of polypeptide chain that are used to get synthesized a functional enzyme. But all these polypeptide chain genes are present on the DNA itself, right? So obviously the DNA is a larger molecule than the enzyme. Next question is here. What would be the molar concentration of human DNA in human cell? Consult your teacher. So with the help of your teacher, you have to find out the molar concentration of human DNA in a human cell, right? Let us see the answer. And this question is coming from or based on the NCERT section 9.1. Let us see the answer. The molar concentration of the human DNA in a human diploid cell is as follows. As you all know, the human body is diploid. Only the gametes that is formed in the reproductive organ, they are haploid, right? So you have to find out the molar concentration of human DNA in human diploid cell. So total number of chromosomes into Avogadro number. And you all know that what is the Avogadro number? 6.03 into 10 to the power 23. This is the Avogadro's number, right? So if you multiply with the total number of chromosome, then you get the molar concentration of human DNA. So 46, human is having 46 chromosome, and you multiply it with the 6.023 into 10 to the power 23. So what you get? 2.77 into 10 to the power 20 moles. This is the molar concentration of human DNA in a human cell. And in this way, you can find out the molar concentration of DNA in some other organism, because other organism, they are having the different number of chromosomes, right? So hence the molar concentration of DNA in each diploid cell in the human is 2.77 into 10 to the power 21 moles. Let us move to our next question. Do eukaryotic cell have the restriction endonucleases? Justify your answer. Now, We are having the two different types of cells, prokaryotes and eukaryotes. Prokaryotes is found in the monerans, that is, for example, bacteria, cyanobacteria, and eukaryotic cell, which is having the true nucleus. So you have to find out whether this eukaryotic cell is having the restriction into nucleus, right? So this question is again coming from the NCERT section 9.2.1. Let us see the answer of this question. No, eukaryotic cells do not have restriction endonucleases. Restriction endonucleases, these enzymes are primarily found in prokaryotes like bacteria. And you all know that one of the example is the EcoR1. EcoR1 means the enzyme that is found in Escherichia coli, right? So where they act as a defense mechanism against invading viruses. So wherever these restriction endonucleases are present in the bacteria, for what reason? They are actually present for the defense mechanism to get rid of these viruses which are attacking the bacteria. To invade these viruses, they have this restriction enzymes. So here you can see one of the way to protect the DNA, that is methylation process, right? They are having the methylation. So this is one of the way. Let us see the answer. So this is because the DNA of eukaryote is highly methylated. Yes, they are highly methylated by the modification enzyme called methylase. Now this methylase put a methyl group to a specific this nitrogenous basis, right? So it protect the DNA, right? Methylation is actually a crucial for regulating the gene expression, development, and maintaining the genome stability. So in the case of eukaryote, they are maintaining the genome stability. They do not get randomly cut by the different restriction enzymes. So the DNA get methylated for the protection in case of eukaryotes. Now, methylation protect the DNA from the activity of restriction enzyme. If the restriction enzyme is present also, then also they will not be able to cut the DNA because of this methylation. So thereby eliminating the need of restriction enzyme. So this restriction enzyme is of no use in the case of eukaryotes, right? So this is the reason that the restriction endonucleases are not found in the case of eukaryotes. A very, very important question, because they are of no use. If they are present also, then also they will not be able to cut the DNA, because DNA got already protected by the methylase enzyme, and they add methyl group. You can see this position, right? They are having the, wherever the yellow is there, yellow position, you can see there is a presence of methyl group. So it protect the DNA. Next question. Besides better aeration and mixing properties, what other advantages do the stirred tank bioreactor have over shake flask? So there are the two different methods, right? Two different methods. And so there is a comparison. The comparison between the bioreactor, which is used for the broad scale, and which is one that is used in the laboratory for the used as a shake flask, right? So this question is coming from the NCERT section 9.3.5. Let us see the answer of this question. So the shake flask method is used for the small scale production of the biotechnology product in the laboratory. So if you are testing, or if you are doing any biotechnology process and you want to get any product, right? So that you do in the laboratory, right? At a very small scale, right? So when you are doing this, taking out any product from based on the biotechnological application, then you take this shake flask method. You can see the diagram of this shake flask method later on. On the other hand, the stir tank bioreactor are used for the large scale production of the biotechnology products. So this is already done a laboratory test, and when the test is passed out and you know that by this procedure you can get this product at a larger scale, then you take it to the bioreactors, right? And there you produce the product based on the biotechnology on a larger scale. So here you can see the diagram. This is the bioreactor. Here you get the product on a very large scale, and this is a shake flask. You shake it because in the bioreactor there is a stirrer that continues moving and producing the product, right? But in the case of shake flask, you have to shake the flask continuously with your hand, right? And that produces the biotechnological products. So this you produce in the laboratory for doing any test, whether you get the product or not. And once you get the product, now you got ensured that by this procedure you can get the product at a larger scale, then you go for the bio reactors. Now, stirred tank bioreactors have several advantages over the shake flask. What are the advantages? Let us see. Small volumes of culture can be taken out from the reactor for the sampling or testing in between the operation, right? You can take out the small volumes of the culture, and that can be taken out for the reactor for sampling, whether the product is of the good quality or whether that can be used as a commercial basis or not, right? It has a foam breaker for the regulating the foam. So it produces the foam, and inside that machine there is a foam breaker. So it has a control system that regulate the temperature and pH. Yes. The bioreactor, as it is used for the larger scale, it is having the control system. Control for what? Control for the temperature, control for the pH. And by the using this control on the temperature and pH, you can increase the product. But what about the shake flask? There you cannot control the temperature. There you cannot control the pH, right? So these are some of the advantages, right, of the stirred tank bioreactor over the shake flask, right? So here you can see the bioreactors, the simple stirred tank bioreactor, right? Here you can see there is a acid base or pH control. There is a temperature control. You can control the aeration also, right? And the second one is the sparge stirred tank bioreactor, through which the sterile air bubbles are sparged. So these are used for the production of the product at a larger scale. Next question. Collect 5 examples of the palindromic DNA sequence by consulting your teacher. Better try to create a palindromic sequence by the following base pair rules. So you must understand what is the palindromic DNA sequence, because there are so many restriction enzymes that once they cut the DNA, you can see those palindromic sequence on the this restriction sites. Okay. So let us see the answer of this question. This question is coming from the NCERT section 9.2.1. Palindromic sequence. Let us understand this. There are certain nucleotide sequence in DNA. And these sequences read the same forward and backward on the complementary strand. Now you all know that there are the two strands in the DNA, right? The ones that runs 5 prime to 3 prime and another that runs 3 prime to 5 prime in opposite way, and they are complementary to each other, right? So they are having the sequence, some of the sequence that are read from 5 prime to 3 prime forward, and in the second strand they are present, same sequence is present in the backward direction, right? So those sequences that can be read from forward and backward same, then it is called as a palindromic sequence. The example which is mentioned in your NCERT book is the Malayalam word. If you read the Malayalam from starting to end and from end to start, you will spell the same thing. So those type of sequences are called as palindromic sequences. So that means the sequence read the same on the two strand. If you read it from 5 prime to 3 prime or 3 prime to 5 prime direction. Now, what is the importance of this? What is the importance of presence of these palindromic sequence on the DNA? Let us understand that. They are the site for the action of restriction enzyme. Many enzymes, restriction enzymes, they cut the DNA at these palindromic sequence. It means their restriction site is, they choose the site where they can cut, and that is very specific, that do not get changed from time to time. It is always be the same for one particular restriction enzyme. And most of the restriction enzyme, they read the palindromic sequence, they choose the palindromic sequence, and there they cut. Their choosing is very specific, right? So they are the site for the action of restriction enzyme. Most restriction site in the DNA are palindromic sequence. So these sequences, it is read from 5 prime to 3 prime or 3 prime to 5 prime. It will be the same nitrogenous base, right? Five examples of palindromic sequences. Let us see the example. This is very important. So here you can see palindromic sequence. First one, 5 prime to 3 prime, GAATTC. If you are reading this, now read this way, GAATTC. This way you are reading, right? So it is giving the same sequence, right? And this is the respective enzyme. This is the sequence of the EcoR1 restriction site. So EcoR1, wherever it get the GAATC, it will cut at that particular point, right? Now, second one, 5 prime GGATCC, right? Now here you see, just opposite, the second strand, GGATCC, right? So if you read Malayalam this side or Malayalam this side, it will be the same. Similarly, these nitrogenous base arrangement on the the segments of, not the segment, strand of the DNA will be the same, right? So this is the restriction site for BAM H1 restriction site, BAM H1, right? Now see the third one, 5 prime AAGCTT. And similarly, you see reading this from opposite direction, AAGCTT, right? Very simple. And this is the sequence for the Hind third, means this is the third third enzyme that was discovered in the this particular bacteria. Fourth one, GATATC, and this is, if you read from this direction, GATATC. This is for eco R5. Eco R5. This is the fifth restriction enzyme that was discovered in the E. coli bacteria. Fifth one, 5' to 3', CTGCAG, and this also read in the same way, CTGCAG. This is the PST1 restriction. Next question. Can you recall meiosis and indicate at what stage a recombinant DNA is made? Now they are asking the recombination. In the case of meiosis, there is a recombination process goes on. So at what stage this recombinant DNA is made? A very important question. This question is related to the cell division chapter also, right? So this question is coming from the NCERT section 9.3. Let us see the answer of this question. Meiosis. It is a type of cell division, right? Meiosis is a type of cell division. And where does it take? It takes at the time of gamete formation in the case of human. It involves the reduction in the amount of genetic material inside the cell. So what is the reduction of genetic material? Reduction means the genetic material becomes half. And you all know that the DNA means the chromosomes are present in a paired form, right? In the diploid cell. But when it becomes haploid, the chromosome that are present in the paired form, they become single. And in that way, the genetic material inside the cell becomes half. And that is why it is called as reduction division. Meiosis is also known as reduction division. So here you can see homologous chromosomes. One is coming from the male and another is coming from the female. So there is a crossing over. Now here you can see the segment of the blue chromosome get cut and the red one also get cut, and there is an exchange of this material. Now when they get separated at the time of cell division, now here you can see this is a new recombination. This is a new recombination of the genetic material, and that is why it is called as recombinant chromatids. And this process is called as chromosomal crossing over. Due to the crossing over, there is a exchange of genetic material. So the stage packaging of prophase 1. This is the stage the crossing over of the non-homologous chromosome take place. Homologous chromosomes are present in one organism only, but in the non-homologous chromosomes, right, there is a stage that is called as this meiosis only. Prophase 1, there is a packaging stage. So the crossing over involves the exchange of the segment of DNA between the non-sister chromatid of the homologous chromosome. And the result is of the recombinant DNA. In the previous figure, we have seen there is a formation of the recombinant chromatids, and that is called recombinant DNA. The last figure you can see recombinant chromatids. So earlier it was homologous chromosome, then chromosome crossover is there, and after the crossing over there is an exchange of genetic material, and that gave rise to the recombinant chromatids. Next question. Can you think and answer how a reporter enzyme can be used to monitor transformation of host cells by the foreign DNA in addition to the selectable marker? This question is coming from the NCERT section 9.2.2. 2? Let us see the answer of this question. So, reporter enzyme, it is encoded by the reporter gene in the vector. Now, you must know what is the reporter gene. So, reporter gene in a vector is a tool that is used in the molecular biology to study the gene expression and regulation. Whether the gene is expressed or not, to study that, these reporter gene are present. If you want to do the recombinant DNA technology, so your result is successful or not, for that you need a reporter gene that reports you, okay, your gene which you want is successfully got incorporated in a particular organism, right? So that is present in the vector. So a vector should have this reporter gene. Okay, so what is reporter enzyme? It is encoded by the reporter gene in a vector. Where this reporter gene is present? It is present in a vector. This is often used to find out successful transformation of host cell. You incorporate a DNA inside the host cell, so there may be a chance that this particular DNA get incorporated into the host cells, but there may be also a chance that this DNA get lost and they do not get incorporated into the that host cells. There could be a chance, right? Not 100% result you will get that every cell is getting that successful incorporation of the foreign DNA. No, it never happens. So you have to take out those transform cells. Transform cells means in the one in which the DNA gets successfully incorporated into the host cell, right? So how to know that this particular cell got incorporated? So here you can see blue-white screening. You can see there are some colonies of the microbes in the figure that are blue in color, whereas some are white in color. So example is the LAC-Z gene. This LAC-Z gene encodes the beta-galactosidase enzyme. There is a LAC-Z gene in the LAC operon that encode for the beta-galactosidase enzyme. Now this enzyme cleaves X-gal to produce a blue product. And this coloration report that the integrity of the transformation of the host cell. So if the cell get transformed, it turns to the blue color. It get transformed to the blue color. And this coloration report that this particular colony of the bacterial cell, they are having the transformed host cells, right? So here you can see this is one of the bacteria, right? This is one of the vector. You incorporated the vector, and this vector you can see the blue colony. Now blue white screening, you can see the once this LAC-Z, which is yellow in color, get incorporated, it changes the product and you get this product. So that is showing the blue colony. Otherwise it will be white colony. In the one in which this vector has not been incorporated, this LAC-Z gene has not been incorporated, that will produce the white colony only. Next question. Describe briefly the following. A. Origin of replication, that is coming from the NCERT section 9.1. B. Bioreactors, coming from the NCERT section 9.3.5. And C. Downstream processing, that is coming from the NCERT section 9.3.6. Let us see the answer. Origin of replication. Origin of replication is defined as the DNA sequence in a genome from where the replication insert. You must have seen that every vector is having the origin of replication. So ORI is a region from where this DNA starts its replication. So every bacteria is having ORI region from which it starts its replication, right? In the same way, right? This vector is also having the ORI region. The initiation of replication can be either unidirectional or bidirectional. So the replication, it can either be start in one direction or in bidirectional. A protein complex recognizes the on-site, unwinds and two-strand and initiate the copying of the DNA. So there is a requirement of some of the protein for the uh, replication, right? And these protein helps to unwind the two strands of the DNA. Once the DNA get unwind, there is a starting of the DNA replication with the help of DNA polymerase enzyme. So presence of ORI in plasmid making them self-replicating unit. Self-replicating unit, it means that they can make their own copy, right? Here you can see this is the origin of replication. You can see the replication bubble is there, right? And this is the parental DNA, parallel. But after some time, at the origin of replication, they can start their DNA. And you can see this maroon color. This is the two new DNA strand formed on the parental DNA template, right? Bioreactors. Bioreactors are large vessels. It is used for the large scale production of biotechnology products from the microbes. We produce some of the biotechnology product by using the microbes, right? Microbes are the one of the very important part in the biotechnology. So bioreactors provide optimum condition to obtain the desired products. So those optimal conditions are optimum temperature, pH, vitamin, oxygen, etc. And you all know that the microbes, that is Saccharomyces cerevisiae, Saccharomyces cerevisiae, that is also called as yeast, that is the backbone for the two very important industries. Liquor industry, in which the beer, alcohol, wine, whiskey all are made, right? And there this Saccharomyces cerevisiae are used. And there is another baking industry, bakery, there also this yeast cells are used. Bioreactors, so they have a different controlling system, an oxygen delivery system. You can control the oxygen, the foam controlling system, the foam that is produced by the continuous circulation of the air. There is a pH and the temperature control system as well. A sampling port of obtain a small volume of culture for the sampling. You can take out a small sample from this bioreactor. Example, stirred tank bioreactor and sparged stirred tank bioreactor. These are the two different types of bioreactors. So difference between the stirred and tank bioreactor and sparged stirred tank bioreactor. The sparge stirred tank bioreactor has a sparger, and the sparger introduces sterile air. The air will not contain any type of microbes or oxygen into the culture medium. The second one is the sparged stirred tank bioreactor. Now, next is the downstream processing. It is a method of separation and the purification of the foreign gene products after the completion of the biosynthetic stage. So after the biosynthetic stage, there may be some contamination that could be present, right? So first of all, we do the separation. You take out all the products, and after that, there is a purification of the foreign gene product. You purify that. You should have only the product, not any other thing. So the product is subjected to the various processes in order to separate and purify the product. So it follows the so many steps in order to get separated the unwanted debris of the substances from the product, and you ultimately get the purified product at the end. So after downstream processing, the product is formulated and it is passed through the various clinical trials for the quality control and other tests. So here you can see, you get the products, and with the product you are having some impurities. So impurities, they get separation. Product and impurities, they get separation. There is a purification product, purification of the product, and after that you get formulation. Means your product is totally free from any other type of contamination or impurities, and there you get the exact product that you want, purified one. So explain briefly. Next question is to explain briefly. First is PCR. This is coming from the NCERT section 9.3.3. So PCR, that is also called as polymerase chain reaction. This is one of the very important tool that is used in the case of biotechnology. PCR is a molecular biology technique used to amplify the DNA. What is the meaning of amplify DNA? To increase the number of copies. See, you all know that you have to take out a foreign DNA from a particular gene that you want to incorporate to your host organism through a vector. Now, you incorporate it into a vector, and at the time of incorporation into the host in the laboratory, it may get lost in the nutrient medium. Then what you will do? You will start the procedure from the starting. You will again take out that organism with the help of restriction enzyme. You cut the DNA fragment, you again take out, introduce it into the vector, and again try to put it into the host genome, right? So this is a very tedious job, right? So what we can do? You first take out the DNA from a desired organism that you want to incorporate into a different organism, a host, you can say. You should amplify that particular DNA. You should make a number of copies of that DNA first thing, right? So here, that is the amplified DNA. It is used to gene manipulation. Gene manipulation means you are changing the gene. Why you are using this PCR technique? Because you want to manipulate a gene. Whose gene? Host genome. So it involves the usage of enzyme polymerase, which polymerizes the DNA. So in this machine, you use an enzyme that is called as enzyme polymerase, right? And that is used for the polymerization of DNA. So that could be called as DNA polymerase. But this DNA polymerase is quite a different from the other cell. Raw material that is used for the PCR technique: primer, deoxy NTPS, that is nitrogenous triphosphate to extend the primer, template strand, and thermostable DNA polymerase that is called as TAC polymerase that you get from a bacteria that is called as Thermus aquaticus. This bacteria can withstand a very high temperature. So steps in PCR, you can see the first step is the denaturation that is done at the 94 to 96 degrees centigrade. At this temperature, the DNA fragment gets separated. The DNA strands separate by breaking the hydrogen bond between the base pairs. The two strands are connected with the help of hydrogen bond. So at this temperature, these hydrogen bonds get easily broken and the DNA strand gets separated. Annealing. This is done at the 50 to 65 degrees centigrade. Primers are bind to the matching the DNA strand. And extension or elongation, this is done at the 72 degree centigrade and makes the new DNA strand by adding the nucleotide to the prime. So here you can see the figure. This is the denaturation at 95 degrees centigrade. You can see the DNA strand got separated. And this red color, the primers got added at both the ends of the DNA strand, right? Basically 5 prime to 3 prime. And the new strands can be formed here. You can see the orange color, the new deoxyribonucleotide triphosphate got attached to it and the DNA strand got fully completed. Restriction enzyme and DNA. What is restriction enzyme? A protein that cleaves the DNA at a specific nucleotide sequence. And they commonly found in the bacterial cells. Where they are present? They are present in the bacterial cells. And they provide the defense against invading viruses. Here you can see the restriction enzyme that cut at a very specific site, that is called as restriction site. And they are present in the bacteria or any other prokaryote because they should get rid of the invading viruses. The example is the EcoR1. It is obtained from the E. coli bacteria and it recognizes the sequence and cuts inside it. The sequence is GAA-TTC that you can see in the figure. DNA, that is also called as deoxyribonucleic acid, that is the polymer of nucleotides, genetic material for all living organisms. Right here you can see the cell. Inside the cell, the nucleus is inside the nucleus, chromosomes are present, and when you open the chromosome, what you find? The DNA. Right, this is the genome. And you can see the plasmid DNA, the small circular DNA that are present in the bacteria as a plasmid, apart from the chromosomal DNA. And mitochondria, inside the mitochondria again you can see the mitochondrial DNA. These are again the plasmid like bacteria, right? So the types of DNA, here you can see genomic DNA. What is the properties of this? Linear double stranded DNA. This genomic DNA is a linear, means straight double stranded DNA, contain genetic information in the organism. What they contain? They contain the genetic information of the organism. Second is the plasmid DNA. Plasmid DNA generally found in the prokaryotes. And covalently closed circular double-stranded DNA beside the bacterial, inside the bacterial species, and beside the circular DNA, there is one circular DNA, that is larger one, that is also called as chromosomal DNA of the bacteria. Beside that chromosomal DNA, there are small circular DNA that is called as plasmid DNA. So they contain some other genes like antibacterial resistance gene. Mitochondrial DNA, covalently closed circular double-stranded DNA present in the mitochondria. Right, mitochondria and chloroplast, they also contain DNA. So there is a mitochondrial DNA in which these small circular DNA are present. Right, okay, so this is all about today's session. Thank you very much for attending today's session. We will meet again same time. Till then, take care and bye.